The following is an original production of AZPM News.
Arizona Science is supported by Research Corporation for Science Advancement.
For Arizona Public Media, I'm Leslie Tolbert, Regents Professor Emerita in Neuroscience at the University of Arizona, and this is Arizona Science. Our guest today is Dr. Andrew Capaldi, professor in the university's Department of Molecular and Cellular Biology, with joint appointments in genetics and in the Bio5 Institute. His research team is making really exciting breakthroughs in understanding the molecular machinery inside our cells. Andrew, thanks for joining us.
Oh, thanks for having me. It's great to be here.
Before we get to research, tell us a little bit about your background. What field did you get your PhD in, and what drew you to what you're doing now?
Oh, sure. So when I went to college, I was interested in math and physics, but my dad's actually a professor of molecular biology. So in the background, I kind of always knew that's what I was going to do. And then I ended up working in biophysics. And then while I was doing my PhD, a new field emerged called systems biology, which was mathematics and network level analysis of cell function. And as soon as I started reading those papers, I knew that's what I wanted to do. So I went to do a postdoc at Harvard and learned all about that, and then ended up here 4 years later.
So your research program focuses on molecular signaling that goes on inside our cells, controlling basic cellular behaviors like finding nutrients or deciding whether to grow, how fast to grow. What's the big picture question that you're asking?
Inside all eukaryotic cells, so from yeast, which we focus on, to plants, to invertebrates, to humans, there's one protein complex called the target rapamycin complex 1 that acts as the central computer that tells the cell how much should I grow and how do I balance all of the pathways that need to support that growth. And so 25 years on, the community has discovered 80 different proteins that control this protein complex. It has to be precisely balanced at all times. And so the evidence for how important that balance is when TORC1 is too active, that leads to hypergrowth. So this is controlling the growth of the cell. That hypergrowth leads to cancer. It can lead to epilepsy, a variety of overgrowth disorders. If the TOR activity is too low, then it leads to neurological disorders. It can also even lead to clinical depression. So there are many excellent groups around the world studying the molecular mechanisms of TOR regulation. But what we're really interested in is how is that complex precisely balanced with so many regulators to control hundreds and hundreds of outputs to let the cell set its growth rate.
Tell us more about the key components of the signaling systems you're talking about.
Yes, so we're really interested in cell growth control, and the central regulator of cell growth control in all eukaryotes on Earth is called the target of rapamycin complex 1, or TORC1. And so that's really, we think of that as the central hub of the network that we study. And many of our experiments are designed to really understand how it works, and then in other cases, how it works in combination with other key signaling pathways.
And how many sites are there on this molecule for binding? I mean, what's the mathematical extreme of possible interactions
That's a really
with this molecule?
great question, because sometimes that boggles my mind, thinking like, how can there be this many regulators? So it's a very, very large protein complex, almost as big as a ribosome. And it docks onto different regulators in different conditions. And in general, at least 10 or 20 regulators are bound in one condition to modulate the way this thing works.
So you use genetics and biochemistry, microscopy. How do you pull these threads together?
Yeah, so we use a lot of approaches because sometimes we have to drill into the details. But I think what makes our lab somewhat unique is we really focus on these large-scale systems biology approaches. So the approach that underlies a lot of what we do is, instead of just looking at one or two molecules in a cell, we use techniques where we can measure 10 or 20,000 molecules simultaneously in a cell. And then we put the cells under different conditions that would regulate their growth. And then we do that in mutations, where we introduce mutations that alter the TORC1 signaling network and ask, how does that change the output across the entire cell?
And the mutations that you induce are things that you've seen in nature, or is this just a way of tweaking this gene point by point by point to figure out what each
So that's
part
a good
does?
question. Sometimes there are things seen in nature, but mostly what we're doing is making pinpoint mutations that break one wire in this network. So what we're interested in doing is we view this as a large signaling network. And so we want to break the wires in this network and then ask, how does its function get interrupted or disrupted? And so, for example, in our most recent study, we used a technique that can follow 20 to 30,000 chemical modifications at a time across a cell. And then we made these pinpoint breaks, taking out proteins or making point mutations in proteins in the network. And then we asked, how do these 20,000 changes alter in the face of breaking that one connection? And so sometimes this is referred to as reverse engineering. So if you knew there was a wiring diagram and you could cut each wire and then ask how the circuitry changes, you can back calculate how this thing works together.
So tell us more about the molecular biological techniques you're using to make all these mutations.
So we work in Saccharomyces cerevisiae, which is the budding yeast, the same thing used to make bread or wine. And we love that because it's the simplest eukaryote. So it has over half of its proteins are the same as humans. The TOR signaling circuit was discovered in yeast and is very similar to humans, but it's very easy to make precise mutations. So we introduce pieces of DNA that have the mutations on them, and the yeast then actually cuts its own DNA and replaces the DNA with these mutations. So at this point, last time I looked, we have something like 4,500 mutant strains we've built. So we can cut anywhere in the cell. We can introduce tags to pull molecules out. So it's very, very easy and fast in yeast. And one of our first studies in yeast, we actually studied TOR signaling in 6,000 different mutants using robots, and then reconstructed the network from asking what every single gene does in the network. So yeast is a great genetic tool for picking apart these kind of networks. In the field of systems biology, a lot of the initial studies start in yeast. And the reason is while it has so many components conserved and similar to humans, it's also just a lot simpler. So as you get more and more complex organisms, they have many versions of genes, many redundant genes. And so doing the kind of study we're doing gets much more complex, because instead of just eliminating one gene, sometimes you have to eliminate 4 or 5 genes. And instead of measuring 20,000 events, you need to measure 100 or 200,000 events, which often doesn't work well because the technology isn't quite there to see that level of complexity, yet it will be soon. But the other really important thing for us is we're trying to understand how cells grow. And a yeast is a free-growing organism, and we can put it in all different conditions, and it's realistic. The studies that are going on with human cells are still quite unrealistic because very few cells grow outside of a tissue. And they actually won't continue growing for very long. So you usually have to use a cancerous version of a cell. And that means you've already messed up the TOR pathway we're studying in the first place. So it gives us this simplified framework, and that will help us understand how to ask questions later in human cells. But it's also, at this point, technologically attackable, whereas human cells are just on the edge of that.
Are people, especially in the medical field, taking up some of your results and starting to use that way of thinking in treating human disorders yet?
That's our hope, and we really think especially this newest study will eventually lead to that. So one of the really interesting things about TOR that's been very confusing is mutations in different upstream regulators, so the different controllers, lead to different diseases. So some of the mutations lead to cancer, some of them lead to epilepsy, some of them lead to clinical depression, some of them lead to diabetes, some of them lead to tumor diseases that are not cancerous. So the most famous is called tuberous sclerosis. And so the question has been, why would different mutations lead to these different diseases? And so we think this multi-layered regulation we've uncovered explains that.
Well, thanks very much for this glimpse into some very, very exciting research. Andrew Capaldi is a professor in the university's Department of Cellular and Molecular Biology and a leader in unraveling and trying to understand the inner workings of cells, the fundamental building blocks of life. You can listen to this and all Arizona Science conversations at azpm.org/Arizona Science. I'm Leslie Tolbert.
Thank you to Research Corporation for Science Advancement for their support of Arizona Science.
This has been an original production of AZPM News.